Hello everyone, we're just going to, hopefully you're enjoying your snow day. Will be the last lecture for the exam on Friday, 3/9. All right, so exam, and this lecture covers through chapter 22.3. All right, so the exam is on 20, 21, and 22 up to 22.3. Okay, so on Monday, our last class, we were left off talking about the cells of innate immunity. And remember that innate immunity is the immunity that you are born with, right? And this is non-specific. Okay, so these cells of the innate immunity, the cellular responses of innate immunity will attack any type of cell. So this is unhealthy or unwanted cell. It doesn't matter if this is your own cells or outside, or what we call non-self, right? A non-self cell. So this cell, this natural killer cell, for instance, will run into a cell that they recognize. It has a difference to them. It is not wanted, and so the cell will induce apoptosis by releasing these little guys called perforin. And perforin, what it does is actually binds in here, and then actually makes a little hole, makes a little tunnel. Okay, that little tunnel, then this granzyme goes in through the tunnel and then causes the cell to induce apoptosis. And it doesn't matter which cell it is, the natural killer cell will run into whichever one that's there. Okay, other cellular defenses include eosinophils, which attack multicellular parasites, and they do something called degranulate. So they don't forget that this eosinophils are a granulocyte, right? So they have cytoplasmic granules. Okay, and those granules are the enzymes and stuff. So, right, so those eosinophils, when they run into a cell, they degranulate. They release their granules, and then they release proteins that form transmembrane pores in the parasite cells, similar to what you've seen with perforin. Okay, so the cells of the innate immune system, eosinophils, the natural killer cells, the neutrophils, they recognize microbes based upon foreign because of receptors. So on the cell surface, there's receptors, right, such as toll-like receptors that identify the cells as what they are, right? So they bind to, natural killer cells will bind to the cell and then influence release of the granules and cause apoptosis or breakdown. Okay, so here's an eosinophil, right, which are not very numerous in the blood, but they run into this parasitic worm, and they recognize from the receptors that are on this worm and on the cell surface, right, that this is not self, this is foreign. Okay, if this is foreign, then it releases those cytotoxic chemicals, and that breaks down and kills off the parasitic worm. Okay, other non-specific immunity or actions are by antimicrobial proteins, right? So these are not necessarily released by specific cells, right? They're present. These antimicrobial proteins are molecules that function against microbes, and so they'll function against the microbes, and they will be present before the microbes get there, right? One class are interferons, okay? And the way interferons work is that they are released by a viral infected cell, All right, so here we have a cell. Okay, here's a cell, happy as can be. All right, for you, this is a self cell. All right, and a virus comes, infects it. I'm just going to draw a little virus, infects the cell. Okay, and the way viruses are is that they have to hijack the cell to produce more little viruses. Right, and then those viruses get released. But before the cell does that, before the cell recognizes that it's been infected by the virus, and before it is hijacked completely, it starts to release interferons. Okay, so the interferons, IFN, alpha and beta, okay, are released. And when they're released, what they do is the interferons work on neighboring cells. All right, so these interferons will work on this one, and on this cell, and this cell. So when the virus starts to overtake the cell, and these virus particles are produced, right, it actually stops the adjacent cells, right, the cells nearby, from being infected by the virus. So in order for a virus to attach to the cell, it has to attach to a specific receptor. So the interferon comes in and maybe it makes the receptor be internalized so a virus can't attach. It prevents the spread of viruses. Okay, and then it also stimulates natural killer cells to come by, right, and kill this cell so that the virus can't be propagated from one place to another. These cells, it stops. All right, so this is kind of what it looks like, the effects of interferon. All right, so here's an infected cell. It releases interferon, and then that causes, synthesizes enzymes that interfere with viral replication. So now this cell can't produce more virus. This cell causes apoptosis, and it triggers that to happen, right? Perforin, all right, and there are all whole steps of ways to prevent the propagation of the virus, because the virus has to hijack the cell, produce more viruses in order to continue reproducing. Another antimicrobial protein is what we call the complement system. Complement is a group of proteins that are present in your blood plasma. Okay, so what they do, they're synthesized by the liver, and what they do is they will be initiated by some type of event. All right, so there's a couple pathways. We call the classical pathway or the alternative pathway. All right, so classical pathway, antibody binds to a foreign substance and then complement is activated. All right, the alternative pathway, complement binds directly to the outside of a bacterial or a fungal cell wall. All right, so the classical pathway is what we call is antibody mediated. Okay, because we need the binding of the antibody first, right, and then complement is activated, or the alternative pathway, complement can do it on its own. All right, this is a great system against bacterial infections. All right, so what it does, so here's complement over here, so we'll go over here. Complement, if you look, there's no antibody that is present here, so this would be, an alternative pathway. Right, and in this case, the complement proteins bind to the polysaccharide of the pathogen, and they form a complex called a MAC protein. Right, a MAC protein, a membrane attack complex. That's what MAC stands for. Right, membrane attack complex. Right, and if you disrupt the integrity of the membrane of the cell, right, that would cause the cell to lyse. Right, that's why we call cytolysis, cell breakage. Okay, and then over here you have an antibody-mediated pathway. So this is a classical pathway for complement activation. So complement, antibody, antigen, right? So in this case, this is an erythrocyte. This is an elimination of immune complex. So an antibody binds to, in this case, an erythrocyte, or would be a bacteria or something of that nature, and the complement is activated, and then it brings it somewhere to be disrupted. So, all right, the cells move to the liver where their complements are stripped off and then cleaned out. Okay, so we have complement, interferon as antimicrobial proteins that are used to prevent pathogens from getting in and infections from spreading. Another non-specific reaction, this is really an inflammatory response or an immune response, is inflammation. Okay, so inflammation is an immediate response. This is response by the body as a way of warding off pathogens. Okay, this is a local, non-specific response of vascularized tissue. All right, to injury. So vascularized tissue, that means we have vessels. All right, so vessels, and this is blood vessels. So the blood vessels are going to change in behavior in response to some type of injury. This is part of innate immunity. Why is it part of innate immunity? Again, it's non-specific, okay? And it was present from the beginning. So what you have is during inflammation is the tissue around vessels is injured in some way, whether it be mechanical injury such as a scrape, excuse me, such as a scrape or a breakage in the skin, or another pathogen injury, such as a viral infection or bacterial infection, really. Okay, the injured tissue changes its behavior in response to the injury. Okay, so the injured tissue and the other cells of the innate immunity, basophils, mast cells, and then also the infectious organisms with themselves, or what we actually would call, could be caused by the breakdown of these organisms, right? So it could be like remnants of the pathogens themselves, cause and release chemicals that initiate response. These chemicals are called pyrogens. Or actually, no, sorry, that's wrong. All right, the chemicals initiate the response, though. All right, so we'll talk about pyrogens in a minute, but that's not what we're talking. So histamine, leukotrienes, prostaglandins, chemotactic factors, these chemicals induce vascular changes. Okay, so vasodilation, that means increased blood flow. All right, more blood to the area. Increased capillary permeability. That means increased fluid out of the vessels. All right, and increased endothelial expression of molecules for leukocyte adhesion. So what that means, we're going to increase the ability of white blood cells to go to that area. Right, they're going to go to that area of tissue damage. So when we think, I want you to remember these things. So increased blood flow, increased fluid out of the vessels. And when we talk about the signs of inflammation, we're going to go back to those. Okay, so now we have recruitment of leukocytes. So the tissue has released these chemicals that have induced the ability of white blood cells to adhere to that area, so they get stuck. They go flowing by the vessels. There's more blood flow, so more white blood cells go flowing through, and then they adhere, so they get stuck there, right? So they get stuck to the endothelial cell adhesion molecules, and then they do. Remember, we talked about diapedesis. This is the way that these white blood cells escape, and now those blood cells have become increased permeability. They've become more leaky, so it's easier for them to get out. And then we have this thing called chemotaxis. Chemotaxis is an attraction, right? So leukocytes migrate toward chemicals released from damaged, dead, or pathogenic cells. So, we have a blood cell, right, and we have a bacteria that's out here releasing something, okay, and now we have a white blood cell that is floating through here, and it's going to squeeze its way out, and then it's going to move towards these chemicals, right? That's called chemotaxis. It's being attracted towards something, right? So it's going to go seek it out, all right? And so leukocytes also release the cytokines, which then stimulate the production of more white blood cells, and then macrophages may release pyrogens. Usually inflammation is really a local thing, right? You think you get remember you scrape your knee, you get a bruise, something, you get hit with a baseball during practice or something, you get inflammation, you get a bruise, a swelling that occurs. And it's red, swelling, hurts, right? But it's usually pretty local. It's not. This macrophages is more of a systemic thing, okay? So with all that extra blood flow and extra permeability, you have more immunoglobulins, clotting proteins, and these things called kinins, right? So these stimulate pain receptors. So all of this also further increase capillary permeability, right? So all of this is kind of still moving through, right? End up with some effects of this. So with increased permeability and vasodilation, you end up with more fluid in the tissue, right? So it moves from the blood to the injured and infected area. So out of the blood cells, right, we get fluid moving out of the blood into the tissue. Okay, what does that cause? That causes swelling. All right, it also increases pressure within the tissue and forces more fluid into the lymph system. All right, so now pick up bacteria and eventually go into the lymph nodes, right? And then it will also expose the bacteria to immune cells. All right, this is all in an effort to start the immune process. All right, you fall off your, So here's your effects of inflammation, right? So first, you fall off your skateboard and scrape your knee, you get some bacteria in there, and now bacteria recognize, and you end up with release of inflammatory and chemotactic factors. The mast cells recognize that, and they start to release all of these histamines, which will increase vascular permeability, right, and recruit more white blood cells, right? So you get cell adhesion molecules. Now they get stuck here. They diapedese through this area. Vascular changes. You get increased blood flow and capillary permeability. So flow, blood's moving faster, and fluids moving out easier. And then recruitment of those immune cells, margination, diapedesis, and chemotaxis. So attachment, squeezing, a release from the vessel, and then attraction towards where these chemicals are, and then you get delivery of plasma proteins that also increase permeability, right? But what you have here now is fluid is released from the blood, and that exudate ends up going inside the lymph cells and then exposing that increased uptake by the lymphatic capillaries and exposing the bacteria to the immune system cells, which are housed within the lymphatic system. Okay, and so what you end up with are 4 cardinal signs of inflammation. Redness, right, which is from the increased blood flow, right, from vasodilation. You have heat, also has increased blood as a result of increased blood flow, and also increased metabolic activity within the area. The cells in the area are already starting to heal. So their metabolism goes up, they're breaking things down, and they're starting to move towards that. You get swelling due to increased fluid, what we call edema, right? That we know what that is. and then pain from the stimulation of pain receptors. So redness, heat, swelling, pain, and then that's the cardinal signs of inflammation. And then loss of function could happen, right, from pain and swelling in severe cases or prolonged, especially prolonged inflammation. Okay? So that's very chronic inflammation would be that prolonged stuff, right? So during the duration of the acute inflammation, about 8 to 10 days, I'll tell you, if any of you remember that I had a elbow inflammation. It was swollen, it was red, it hurt. I had a little bit of a loss of function because of the swelling, right, especially in joints, and that was from bacteria just getting in past the skin, and I had to take antibiotics for it. Eventually it goes away, but it's a way of your immune system attacking those potential pathogens. Okay, a more systemic response, right? So this is more systemic, meaning the whole body rather than a local response of inflammation. A more systemic response is pyrexia or fever. Fever is characterized by a elevated body temperature, at least 1 degree Celsius or more from normal, which is 37. Okay? And this results from a release of those chemicals, what we call pyrogens, right? Such as interleukin 1. Pyrogens are chemicals that induce elevated temperatures. Okay, and really this is a body's response to an infection, a way of systemically trying to block and stop the infections from spreading. Okay, so the pyrogens are released. They circulate throughout the blood and target the hypothalamus, which has your internal thermostat. When that happens, that elevates the temperature. Okay, it raises the temperature set point, leading to fever. So you go from 98.6 to 100. All right, 100.1 or 101 or whatever, up to nothing more than let's say 104. All right, so fever has 3 stages: onset, stadium, and defervescence, which comes back down. So onset, the temperature begins to rise. Your hypothalamus is stimulated by the presence of pyrogens, and the hypothalamus releases hormones to stimulate the dermis blood vessels to constrict, less heat loss, right? So typically signs of fever, someone has a white color but their skin's really warm. Okay, the shivering, right? They get the shivers, may in response to some chills, and so what ends up happening? The temperatures begin to rise. So you constrict the vessels, you conserve heat, and temperature goes up. So it goes up to 101 degrees Fahrenheit, we'll say. And then we reach this plateau, which is stadium. This elevated temperature is maintained, so it stays up that way. Your metabolic rate increases to promote elimination of the harmful substance that's doing this, whether it be a virus or bacteria. A lot of times a virus are the culprits of fevers. Okay, and then your liver and spleen, they bind some chemicals to help slow microbial reproduction. So zinc and iron will be retained, will help to prevent microbial reproduction from growing. And the elevated temperature also helps with this. Okay, so if you were to grow bacteria in a lab, in a little, you can grow them in a little petri dish, in a little Erlenmeyer flask, right, in a liquid culture. You put that in a petri dish. You put that in the oven at an incubator, and it's set at 37 degrees Celsius, which is exactly what human body temperature is. So bacteria have evolved to grow best at 37 degrees Celsius. But if you put them up to 40 degrees Celsius, right, what you end up doing, the growth rate slows. So growth rate slows. And that's equal to fever, right? So fever is a way of increasing body temperature in order to inhibit the growth of bacteria. All right, and so you'll keep it that way until they start to come down. And then defervescence is the time when temperature starts to return to normal. So the hypothalamus no longer stimulated by the pyrogens because the bacteria, the virus, aren't making them anymore. All right, so prostaglandin release decreases, and the hypothalamus stimulates mechanisms to release heat. So sweating, vasodilation, right? So we're coming back down to normal, back down to our 98.6. The benefits, right? So it inhibits the bacteria, the reproduction of bacteria and viruses. It promotes interferon activity, which will help to prevent the spread of the virus, right? Increases activity of adaptive immunity, we'll talk about later on. So now that we have this the initial response by fever, then the rest of the immune system can kick in, and then accelerates tissue repair, and then increases cell adhesion molecules on the endothelium of capillaries and the lymph nodes, right? So that the immune cells can help them do their job. So the recommendation is to leave a low fever untreated, right? So if you kind of have like a little, like 100 degrees, right? That's okay. That's not too 100 degrees Fahrenheit. That's not too high. But if you get like 101, 102, then we have to deal with that, because when you end up going too high, some negative occur, right? So high fever is potentially dangerous, especially if you get up, children in 103, right? Where it changes metabolic pathways and really could cause denaturation of proteins, right? Denature of proteins gets up too high. The proteins they lose their shape, they no longer function, and cause really big problems, especially in the brain, cause possible seizures, and could cause irreversible brain damage, right? Over 106, and death if temperature is over 109, because we can't go back to those proteins being produced. Okay, and so here's our first lines of defense, some pictures that show kind of what's going on, where the innate immunity is, and we picked an infant here because this person's adaptive immunity hasn't developed yet. All right, so these are all the immune response that we have for that we're born with, whether it be in the skin, the mucous membranes, and cells of immunity. Right, and then the second line of defense. So we have our first line of defense, our barrier, and our second line, all the cells and internal aspects that occur within if something gets in past the barrier. Okay, and the last thing I want to talk about is pus and abscesses. Pus is an exudate, meaning it has to do with it's what's left over after the fluid of increased capillary permeability and before it gets in the lymph. That's what's left over, right? It has destroyed pathogens, dead leukocytes, macrophages, cellular debris. It's like this green, sticky fluid, okay? And it's usually, when you see it, it's removed through the skin, right? Or it can be removed by the lymphatic system. If it's not completely cleared, it may form an abscess. So it means its pus is walled off with collagen fibers, and it's kind of stuck in there, and usually requires some type of surgical intervention to remove, or at least to open it, right, to remove the pus. So because we don't want that stuff to kind of just live there, we want it to go away. If it builds up too fast and it's not stuck, we need to kind of remove it, because then the tissue doesn't repair itself. So that covers up the material for our exam on Friday, 3/9, and that goes through 22.3. All right. If you have any questions about this lecture or anything else, feel free to email me. I hope to see you guys tomorrow in lab. And if not, I will see you on Friday. Remember to bring your a pencil and an ID with you to class. See you then.